Okay, everyone, we might get started for today if everyone can hear me. My name's Rachael. For those who don't know me, I'm a postdoc in Dino Giussani's lab over at Physiology, Development and Neuroscience, and I'm happy to be chairing the session for today. So a few things first about just the Cambridge Reproduction seminars in general. So these are hosted by Cambridge Reproduction, and we're really grateful to all of the volunteer coordinators for help organising the series, and of course all of the speakers who have generously agreed to take part. So the seminars are held online on the third Thursday of each month at 1 p.m. This is actually the last seminar of the current academic year, but we will be starting again in the autumn. So if you're a postgrad student or a postdoc in Cambridge and you'd really like to take part in the seminar, we'd love to have you. So I think someone will be posting a link in the chat here. And there's also the Cambridge Reproduction website. So please get in touch and share your research with us. So Cambridge Reproduction is an interdisciplinary initiative, and it brings together researchers from across the university that have an interest in any aspect of reproduction, essentially. So anything from the science, the technology, medicine, or even the arts and humanities aspects. Membership is open to all staff and postgraduate students, and it gets you access to all of our events and funding schemes, as well as regular newsletters. So things like research highlights and opportunities for reproduction research. So to join, all you need to do is access the membership form from the website and send it to Christina Rozek, who is in this session as well. She's the Cambridge Reproduction Coordinator. And again, she can post a link there in the chat for us. And move on to our second speaker now, who is Isabella Inzani. Isabella is a final year PhD student in Professor Suozan's lab at the Metabolic Institute Research Laboratories. And she is doing research investigating the effects of maternal obesity on the health of offspring, looking at developmental programming outcomes. So today she's going to be speaking to us about fetal hypoxia in a mouse model of obese pregnancy. So take it away, Isabella, if you're ready.
Thanks very much. Yes, as Rachel just said, my name's Isabella Inzani, and I'm a final year PhD student at the Institute of Metabolic Science. And I'm here to talk about one of the projects I've been working on during my PhD, which is investigating fetal hypoxia in a mouse model of obese pregnancy. So to start with, obesity. Obesity is a problem which has been growing in prevalence worldwide. So much so that it was declared an epidemic by the World Health Organization in 1997. And the problem has only grown from there. We are now at the point where there are more people in the world who are over than underweight. This map here shows the levels of obesity in adult women across the globe. And you can see that there are high levels of obesity in the so-called developed world. And there are also ever increasing levels of obesity in developing countries as well. And specifically in the UK, obesity really is a growing problem. The 2019 Health Survey for England estimates that 28% of adults in England are obese, while a further 36.2% are overweight. When stratified for age, this means that approximately 3/4 of people aged 45 to 74 in England are either overweight or obese. And the problem is getting worse in every age group, particularly during the pandemic. What's shown here is some recent data which was collected during the pandemic in Year 6 children who are approximately 10 to 11 years old. Looking, for example, at the top line, this shows that the combined levels of overweight and obesity in this age group, and you can see that there's been a slow increase in prevalence from 2006 to 2019. For example, between the school year beginning in 2010 and the year beginning in 2019, there was a 1.8% increase in the children being overweight or obese, from 33.4% to 35.2%. However, in the following year, during the pandemic, there was a 5.7% increase in this single year alone. And the most recent data now shows that 40.9% of Year 6 pupils are either overweight or obese. All this data together therefore suggests that obesity is a large and growing problem in both children and adults. And this has very concerning health implications for the population. As I'm sure you're all already aware, obesity is associated with poor health across a variety of different organs and body systems. This includes being associated with heart disease, liver disease, cancer, stroke, reproductive complications, asthma, depression, and type 2 diabetes, to name just a few. However, my research in obesity focuses on a specific subset of the population, pregnant women. As you remember the map I showed you a few slides ago, this was of the levels of obesity in adult women across the globe. And therefore, as the levels of obesity increase, more and more women are entering pregnancy either overweight or obese. Now, this can have a series of negative health implications for the mother, including decreased fertility and increased risk of pregnancy complications, including miscarriage, gestational complications, and difficulties around delivery. And there are also negative health implications for the developing fetus, exposed to the maternal obese environment in utero. For the fetus, maternal obesity can lead to increased risk of stillbirth and increased risk of metabolic and developmental abnormalities. However, these effects of maternal obesity on the child's health can persist beyond fetal life and into childhood and adulthood. This idea that adverse conditions which affect the development in early life can impact the long-term health of the offspring is known as the developmental origins of health and disease, or DOHAD for short. In the case of exposure to maternal obesity, in the adult offspring, this is known to increase the risk of offspring developing obesity themselves, as well as increased risk of diabetes, high blood pressure, and cardiovascular diseases. However, the focus of the project I'd like to talk to you about today is looking here at the fetus. Given we see these negative health outcomes in the adult offspring, we are looking back at the fetus to try and fully understand the environment of the obese pregnancies and how this impacts the fetus. In order to do this, we use a well-established mouse model of maternal diet-induced obesity. Here, you have young female mice who are fed either a control healthy chow diet or a high-fat, high-sugar diet, which consists of high-fat diet pellets and sweetened condensed milk. Now, a mouse model is a particularly useful tool for us for several different reasons. One is that mice have a relatively fast life cycle and a short gestation length, which makes studies feasible over a reasonable time frame. They are also a model which allows for us to control for other lifestyle and genetic variables, something which would be impossible to do in clinical studies. And finally, another advantage of the mouse is that they have multiple fetuses in a litter, often about 7 to 10. This means that we can study male and female fetuses from the same litter. Of growing importance in our field is the concept of sex differences. This is the idea that the same exposures in early life could have different effects on the offspring's health, depending on if they are a boy or a girl. And this can therefore affect the approach to developing treatments and interventions for the mother and offspring. Therefore, in the mouse, having both male and female fetuses from the same mother allows for us to control for the maternal environment, and it's therefore a valuable tool for us to investigate these sex differences. This model of maternal diet-induced obesity has been well established in our lab, and we've used it extensively to study the health of the offspring born from the obese mothers compared to those born from control mothers. We've seen that the adult offspring of obese dams are programmed to have increased risk of cardiometabolic diseases. This includes increased risk of offspring themselves becoming obese, with increased risk of adiposity and adipose tissue dysfunction, increased risk of developing fatty liver and impaired liver metabolism, increased risk of heart disease with cardiac structure and function abnormalities, increased risk of hypertension or high blood pressure, altered hypothalamic signaling involved in the maintenance of energy homeostasis and feeding regulation, and increased risk of type 2 diabetes with impaired pancreatic islet function and insulin resistance. But what exactly is going on in obese pregnancies to program these offspring cardiometabolic dysfunction? Now, previous studies from clinical data and animal models, including our own, have shown that maternal obesity leads to increased maternal insulin and leptin levels and impaired glucose tolerance in the mothers. This can all affect the fetal in utero environment and contribute to the programming of offspring long-term health and disease. However, another factor, which has been relatively less well explored in the context of obese pregnancy, is hypoxia. Hypoxia is a term which describes when there are low levels of oxygen in the tissues. Previous data from late pregnancy studies in our lab have suggested there is increased oxidative stress in the fetus and placenta, and has also been suggested that the fetus is hypoxic compared to the conditions of normal weight healthy pregnancies. In models specifically of chronically hypoxic pregnancy, there's already extensive evidence that a hypoxic environment during pregnancy can have, in some ways, similar effects to that of maternal obesity on long-term offspring outcomes. with increased risk of offspring developing cardiovascular diseases in adulthood if they're exposed to hypoxia during gestation. Therefore, fetal hypoxia caused by obese pregnancy could be a potential programming factor which could help to explain the effects of maternal obesity on offspring health. We therefore aimed in this project to 1, determine the effects of maternal obesity on fetal and placental hypoxia, and 2, if the fetuses of the obese mothers are indeed hypoxic compared to the controls, to identify any maternal factors which might be linked to the obesity-induced hypoxia. And thirdly, to identify the underlying mechanisms. Exactly what is different in the obese pregnancies which leads to the hypoxic conditions developing. And finally, for all of the above, to determine if there are any results which we see which are the same in both sexes, or if there are any sex differences between the male and female fetal phenotypes. So in my study, we used our mouse model of diet-induced obesity to study the fetuses of these mothers. Young female mice, the future mothers, were fed either the control healthy diet or the obesogenic diet throughout their life, both before pregnancy and during the pregnancy itself. A normal mouse pregnancy lasts about 20 days, but we allowed these pregnancies to progress to approximately mid-gestation at day 14, which we refer to as E14. At this point, we injected the mums with a hypoxia marker called pimonidazole. We then collected fetal and placental tissues for analysis. So to start with, the mothers. In all the graphs I'm going to show you today, you can see the control group in black and the obese group in red. So on the graph on the left, we measured the mother's body weight daily from the start of pregnancy to the end point of our study at mid-gestation. And these dams were on the experimental diet before pregnancy as well as during. And you can see that this means that the dams fed the obesogenic diet, the red line, start pregnancy already significantly heavier than controls. And this difference in body weight between the two groups is maintained throughout the pregnancy. On the right, you can see the dam's fat mass, which we measured non-invasively by TDNMR before pregnancy and weekly throughout pregnancy. So you can see that the dam's fat mass is also increased in the group of the obesogenic diet, both before and throughout gestation. At day 14, at the endpoint of the study, we also collected blood from the dams. We measured blood glucose levels and serum levels of insulin and leptin. As you can see, the dam glucose and insulin levels are both increased in the obese dams compared to the controls, which suggests a gestational diabetes-like phenotype. Leptin is a hormone which is released from the adipose tissue, and as might be expected by the significantly increased fat mass you can see in these mice in the graph above, leptin is also greatly increased in the serum of the obese dams compared to the controls. So once we had confirmed our model that the obese dams were indeed definitely obese, we moved on to study the fetuses. And as I mentioned before, the importance of understanding if there are sex differences led us to do all our analyses in both the male and the female fetuses. You can see here on the top left that there are no differences in fetal body weight between the fetuses of control or obese mothers at this time point. However, there is a significant effect of fetal sex, with female fetuses being smaller than the male fetuses in both control and obese pregnancies. A similar result can be seen with the placental weight, which follows the same pattern. There are no significant differences in placental weight between the control and the obese pregnancies, but like the fetal body weight, the female placentas are smaller than the male placentas. The lower two graphs on the slide just show that at mid-gestation, there also don't seem to be any differences with maternal obesity for general litter characteristics, with no differences in litter size or the ratio between male or female fetuses in the litters from the control and obese mothers. Now, if you remember from my previous slide on the model that we're using for this study, 90 minutes before we collected the tissues, we injected the pregnant dams with the hypoxia marker pimonidazole. And importantly, pimonidazole is able to cross the placenta and also enters the fetus. It works as it's reductively activated specifically in hypoxic cells, where it forms stable adducts with thiol groups and proteins. We can then detect the levels of these pimonidazole protein adducts in the fetal tissues, and the level of the pimonidazole detected is directly proportional to the level of hypoxia in the tissues. What you can see here are some sections of whole fetuses that have been cut in the frontal plane, so essentially dividing the front and back. And here you can see the cross-section of several different fetal organs. Outlined in orange on the images, you can see at the top the fetal brain, in the middle of the torso the fetal heart, and just below it, the fetal liver. And these sections have all been stained for pimenidazole adducts. The darker the stain looks, the more pimenidazole is there, and the more hypoxic the tissues are. So when I quantified the stain intensity for the whole fetal section, you can see that there's a main affected group, which refers to the mothers being neither the control or the obese groups. The fetuses of the obese mothers had significantly increased levels of hypoxia compared to the controls. However, there were no differences depending on if the fetus was a boy or a girl. I then repeated this analysis, looking specifically at the fetal organs which I highlighted, the brain, the heart, and the liver. And here you can see the trend persists in the other fetal organs, with an increase in tissue hypoxia in the fetuses of the obese strands compared to the controls. I also performed this same analysis in the placentas. In the mouse placenta, there are 3 main zones, which you can see labeled here. On the inside of the curve, you can see the labyrinth, which is the main site of transfer between the maternal and the fetal blood. On the outside, there's the decidua, which is the maternal side, and between the two, you have the junctional zone. However, unlike the fetuses, when we look at the intensity of the pimenidazole staining in the whole placenta, we don't see any differences in the hypoxia level in the control versus the obese groups, neither the male or the female fetuses. And again, this trend continues if we look at the 3 zones separately. Maternal obesity does not appear to be resulting in increased placental hypoxia at mid-gestation. So once we've established that maternal obesity results in fetal but not placental hypoxia, we looked at the relationship between the fetal hypoxia and maternal factors to try and determine what aspects of the maternal obese environment might be driving the fetal hypoxia. For this, we looked at the correlation between fetal hypoxia levels shown on the y-axis and DAM factors on the x-axis. These were the factors which I previously showed to be different between the control and obese DAMs, namely maternal body weight, fat mass, blood glucose levels, and serum insulin levels. From these graphs, you can see that maternal obesity positively correlates with the maternal body weight, fat mass, and insulin, but not DAM glucose, suggesting that increased maternal fat mass or insulin may be driving the fetal hypoxia in obese pregnancies. We are also trying to understand the mechanisms which might underlie the fetal hypoxia, exactly what differs between the control and obese pregnancies to cause it. One idea is that it is due to differences in the placenta. However, as I've already shown you, the obese placenta itself is not hypoxic at this stage. We have previously shown that later in gestation, the obese placenta shows increased expression of the hypoxia marker protein HIF1 alpha, and that compared to controls, the obese placenta had increased calcification in late gestation. Therefore, it's possible that later on in pregnancy in the obese placenta, they do become more hypoxic compared to controls. However, at this mid-pregnancy time point, we have shown that there's fetal hypoxia present, and therefore precedes any placental hypoxia that might develop later. Therefore, it's not that there is placental hypoxia driving the fetal hypoxia. So we also looked to see if differences in the placental size and structure might impair oxygen delivery to the fetus. However, there were no differences in placental efficiency, which is a measure of fetal body weight relative to placenta weight at this time point. This means it's not that relative to the size of the fetus, the placenta is smaller and therefore has less capacity for transport. We also looked specifically at the mid-placental area of the labyrinth zone, which is shown here on the right-hand graph, as this shows the area most responsible for transport between the mother and the fetus. However, no differences in area were seen. So finally, we looked to see if there were differences in the structure of the labyrinth zone, specifically vascularization, as reduced blood vessels would also impair transport capacity. We did this by staining for the endothelial marker CD31, which forms the lining of the blood vessels, and which you can see in the more zoomed in image of the placental labyrinth zone in the bottom center. However, again, we saw no differences between control and obese groups for either sex. So from here, we moved on to investigate the idea of fetal brain sparing. This is something which is known to occur in chronically hypoxic pregnancies, and it's where, in conditions of low oxygen, the fetus alters blood flow to prioritize blood flow and oxygen supply to the brain at the expense of the other peripheral tissues. However, if you remember from my previous slide, we saw increased hypoxia in the fetal brain for the obese pregnancies, as well as the other organs. Furthermore, if we calculate the ratio of hypoxia levels in the brain compared to the whole torso, we can see there's no difference between groups, which suggests that compared to the levels of hypoxia, comparable levels of hypoxia are seen in the brain as in the rest of the fetus. And finally, we looked at vascularization in the fetal tissues to see if there might be any differences in the perfusion to the fetal tissues and thus oxygen delivery. However, we didn't see any differences in either the fetal heart or liver between groups. And so, to conclude, maternal obesity results in global fetal hypoxia, and this occurs in both male and female fetuses. However, maternal obesity does not lead to any similar changes in placental hypoxia at E14. Maternal body weight, fat mass, and insulin levels are all increased in obese pregnancy and positively correlate with the degree of hypoxia seen in the fetuses. However, we only looked into the potential underlying mechanisms for fetal hypoxia. There were no differences in the size and the structure of the placenta at this mid-gestation time point, and no differences in fetal vascularization, and no evidence of fetal brain sparing in our model. Therefore, the mechanisms underlying the fetal hypoxic phenotype we observe in the obese pregnancy still remains to be determined. And this is just a little diagram that again summarizes everything that I've talked about today, hopefully in a slightly prettier way. Using a mouse model of diet-induced obesity at mid-gestation, we found evidence of fetal but not placental hypoxia. The maternal factors we found associated with fetal hypoxia are down body weight, fat mass, and insulin levels, which are all shown to be increased in the obese dams compared to controls. We've investigated several different underlying mechanisms so far, looking at the placenta, fetal brain sparing, and fetal organ vascularization. However, none of these appeared altered by maternal obesity at this time point. So our search for mechanisms is still ongoing. This is particularly important for us to understand because we know that overall, the obese maternal environment affects fetal development and results in the programming of adult offspring cardiometabolic diseases. If we can understand what differences between obese and controlled pregnancies affect the fetal environment, it can help to guide us in developing interventions to prevent the negative health implications to the offspring. So finally, I'd like to end by acknowledging my lab, and in particular my supervisor, Professor Susan Ozanne, and also Denise Fernandez-Twin, who did a lot of the work for this project. Our collaborator, Professor Dina Ghassani from the Department of Physiology, Development and Neuroscience, and of course, the British Heart Foundation, who support my PhD. So thanks very much for listening, and I think we're now going to move on to questions, so I'd be happy to answer any that you have.